Okay, this is video number 2 on cell transport and homeostasis. We left off talking about diffusion, and we talked about simple diffusion, and the picture you're seeing now is of simple diffusion. We have a solute here moving from high to low concentration through a phospholipid bilayer. You could see here that these molecules are small enough to shimmy through the phospholipids from an area of high concentration to the area of low. And then once they reach equilibrium, you can see that the molecules are able to move back and forth through the phospholipid bilayer as long as equilibrium is maintained. Sometimes, however, you can see these molecules are too large to shimmy through those phospholipids, so they get stuck. However, they still want to move from high to low concentration inside of the cell. So sometimes these molecules need a little help. They need to be facilitated. So what happens is they travel through these carrier proteins, or these carrier molecules, as your vocab would say it, and it's basically like a tunnel where the molecule is able to fit through the membrane from high to low concentration. You're just, like I said, you can't go through those phospholipids, so you need to go through this carrier protein. The word facilitate means to help or to guide, so facilitated diffusion is where the molecules need help going through the membrane. And once again, that help would be that carrier protein. Okay. So what is the difference then between simple and facilitated diffusion? What do you think? It's that facilitated diffusion requires membrane proteins. So they're basically the same thing, except once again with facilitated diffusion, the particles that want to go through are way too big to fit through the phospholipids, so they need to go through that carrier protein. Okay, so now we're going to move on to osmosis, and osmosis deals specifically with water. So just as water flowed from an area of high concentration in the ocean into an area of lower concentration inside the Titanic, for example, water is going to flow through membranes. Now you should picture the penny whistle solo from Titanic. Do 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 do. Do, 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 do. I'll never let go, Jack. I'll never let go. But seriously, osmosis is the movement of water, and we're going through a semi-permeable membrane. So water needs to be able to move in and out of the cell. This is a type of passive transport, so it does not require any extra cell energy. These water molecules are moving with their own kinetic energy, like we talked about in regular diffusion. So a couple things with water and the movement of water. As we know, water is going to pass back and forth through that membrane by osmosis. The direction the water moves in and out of the cell is determined by the concentration of, obviously the water, but we also have to look at the solute. So how much solute do we have? And remember, that solute could be things like salt, sugar, for example. The higher the concentration of solutes in the water, the lesser the concentration of the water, which we'll talk about. And the differences in the concentration of solute versus water creates 3 environments for the cell. You can either have a hypotonic environment, a hypertonic environment, or an isotonic environment. And all of these are actually named after the solute content. Although we're talking about water, they are named after their solute. So our first one is a hypotonic environment. And it's called hypo because it has a low solute concentration. So think hypo, low. They kind of sound like they rhyme a little bit, I guess. So hypo, low solute. So that means high water concentration. So let's see what's going to happen. You can see here we have a beaker. And in that beaker, I've placed 1 red blood cell. And it's obviously larger so that you can see it. And we are in a beaker of water. So the salt concentration inside the cell is 1%. So obviously percentages should add to 100. So if we have 1% salt, our concentration of water should be what? 99. In the beaker, we have 0% salt, so our percentage of water should be 100. So we have 99% water in the cell and 100% outside the cell. Okay, so it's a low solute, high water environment, which would be the water around the cell. So what's going to happen to the water? It wants to move from high to low. So is it going to move in the cell or is it going to move out of the cell? Into the cell. So the water is going to move into the cell, and as that water flows into the cell, what do you think is going to happen to the cell? It's going to get larger. It's going to get so large that it actually could pop, which is called cytolysis, which is one of your vocab words. Think cyto for cytoplasm, and lysis means to break. So cytolysis means cell bursting or cell death, okay? So think about who likes this. Who would like this type of environment? Obviously not a red blood cell. Not good for that. So it's actually not good for animal cells. But it is good for bacterial cells because they have a contractile vacuole. So they need that water to rush into their cells because it provides nutrients. But it doesn't burst because that contractile vacuole can squeeze out that excess water so that it doesn't pop. Plants also like a hypotonic environment because it helps them with turgor pressure. So the water moves by osmosis all the way up the plant and into the plant cells. It fills up that central vacuole, which will then push on your cell walls, and it allows the plant to stand upright or remain rigid. If you were to lose the water from these cells, that's when the plant would actually wilt. So once again, hypotonic environment, good for plants, good for bacteria, very, very bad for animal cells. Okay, so moving on. Next we have a hypertonic environment. So when you're hyper, someone says you're hyper, it typically means you have a lot of energy, right? You have a lot. So with a hypertonic environment, you have a lot of solute. Once again, they are named after their solute concentrations, not after their water concentration. So hypertonic, high solute, which means low water in the environment. So let's look at what happens here. Once again, red blood cell within a beaker. We have our water, we have our cell. Our cell is 1% salt, which means obviously it's 99% water. And our environment has 7% salt, which means it's going to have 93% water. So 99 versus 93. So the high concentration is inside the cell. So the water is going to move from high to low. Where's it going to move? Out or into the cell? Out. Okay, so the water is going to move out of the cell. And as the water moves out of the cell, what's going to happen? The cell is going to shrivel. Okay, which is, not really the greatest thing. This process is called plasmolysis or crenation, not to be confused with cremation, but crenation with an N, okay? And this is not good. You will need to know this for the test, plasmolysis or crenation, either one. So obviously looking at the red blood cell, it is not good for the red blood cell. Plants also would not like this. They don't want to shrivel up. And bacteria also don't want to shrivel up. So all three, plants, bacteria, and animal cells, all hate a hypertonic environment. So hypo, good for some, not good for others. Hyper is bad for everybody involved. So no good for this. Our last environment is called an isotonic environment, and the prefix iso actually means same. So when you're in an isotonic environment, you have the same amount of solute and the same amount of water. So everything's even. So let's check this out. Here's our beaker, 1% salt in our cell, so that's 99% water. 1% salt in our environment, so that's also 99% water. So 99 in, 99 out. There is no high, there is no low. So what's happening? Well, when you're looking at it, nothing is happening. However, we know that this would already be in equilibrium. So the water is actually going to move in and out. You just can't see it happening, and you can't really tell that there's any really change in the cell. But water is moving in and out because we're in equilibrium. And remember, just because you're in equilibrium doesn't mean the molecules stop moving. They continue to move, but they just move evenly, and then they can move in and out as long as equilibrium is maintained. So no visible change. This is the normal cell environment, and this is the environment that maintains homeostasis. So it's good for plants, good for animals, and good for bacteria. Everybody likes an isotonic environment. In the Amoeba Sisters video I was showing you guys at the beginning of this chapter, that's why when you are on an IV drip at the hospital for, to hydrate, you don't want just water going into your cells because that would cause a hypotonic environment. The liquid they give you is actually, that has lots of solutes in it as well, but not too much obviously, but enough that it creates an isotonic environment so that we don't have water rushing into or out of your cells. You want an isotonic liquid so that you can maintain homeostasis. Okay, so looking here, what type of environment are these cells in? Here we had shriveled animal cells or a shriveled plant cell. So that would be a hypertonic environment. Here we have normal blood cells in a normal plant cell, which would be isotonic. And here obviously you can see the cells bursting, and here the word turgid is there, and which means it's very rigid and thick. So that would be a hypotonic environment. Excuse the bell in the background. So let's try this out. If a red blood cell is placed in this environment, the water will move out of the red blood cell. Hypertonic. How about this one? If a red blood cell is placed in this environment, the water will move into the red blood cell. Hypotonic. Good. So all of these things that we've talked about are what's called passive transport. So diffusion and osmosis are types of passive transport because the molecules move with their own kinetic energy and no cell energy, no effort on the cell is put forth. Molecules in passive transport, just to recap, are moved from high to low concentration. Small molecules diffuse through the phospholipids. Larger molecules have to go through facilitated diffusion. They go through those carrier or transport proteins. And once again, we are moving from high to low concentration. That wraps up passive transport. And in our next video, we're going to be moving into active transport. So stay tuned.